Hello everyone, this is a presentation that I gave to the Reactive Metabolites in Diabetes study group recently in their symposium on insulin resistance, new insights and nutritional approaches to therapy. It's on hexokinase 2 linked unscheduled glycolysis hypothesis of insulin resistance. I believe it's probably one of the most important presentations I've ever made. So please keep listening to the end if you can. Thanks so much. The agenda for my presentation is as shown. I'll talk about what is insulin resistance and why is it important. I'll introduce a hexokinase 2 linked unscheduled glycolysis hypothesis. I'll present a mathematical model of hexokinase 2 linked unscheduled glycolysis. I'll look at preclinical and clinical evidence of the hexokinase 2 linked unscheduled glycolysis hypothesis. Then I'll ask, can hexokinase 2 linked unscheduled glycolysis explain functional genomic studies in mice of insulin resistance? I'll ask, what predictions from hexokinase 2 linked unscheduled glycolysis can we make for the clinical setting? And then finally, how can this improved insight guide future improved treatment for insulin resistance? So let's get started. At the outset, let's define what insulin resistance is and say why it is important. I give the definition of insulin resistance of DeFronzo and Tripathi. Insulin resistance is a reduced response of target tissues to insulin compared with subjects with normal glucose tolerance. Important target tissues are skeletal muscle, liver, and adipose tissue. Insulin resistance precedes, some would say parallels, decline of beta cell function and offers an opportunity for treatment to prevent and treat type 2 diabetes. Although 69% of genes linked to risk of type 2 diabetes are associated with beta cell function, as indicated in the pie chart, impaired glucose disposal in insulin resistance increases dysglycemic stress in beta cells, accelerating decline in beta cell mass and function. Insulin resistance also contributes to other morbidities such as decline in liver function, decline in kidney function, and decline in lung function, and other morbidities. We do not currently have a hypothesis that can account for experimental evidence on the development of insulin resistance. Indeed, recently, insulin resistance was said to be a malady without a mechanism. In this presentation, I explore the contribution of hexokinase 2 linked unscheduled glycolysis. In the table shown, I compare the kinetic characteristics, tissue distribution, and other properties of all hexokinases. There's hexokinase 1, hexokinase 2, hexokinase 3, and hexokinase 4, or glucokinase. Hexokinase 1, hexokinase 2, and hexokinase 3 are high affinity glucose binding hexokinases that are saturated with glucose even under normal glycemic conditions. Hexokinase 4 or glucokinase is a low affinity glucose binding hexokinase and has increased activity as glucose concentration increases through the physiological range and into hyperglycemia. Hexokinase 1 and hexokinase 2 are expressed in vascular cells, in muscle and adipose tissue, making them appropriately placed to be involved in the mechanisms of development of vascular complications of diabetes and also insulin resistance. Glucokinase is expressed in pancreatic beta cells and liver, which makes it appropriately placed to be involved in a glucose sensor function. Hexokinases are gatekeepers at the entry into glycolysis with a critical role in glucose disposal and dysfunction in hyperglycemia. Whilst glucokinase has rightly received much attention as glucose sensor in the pancreatic beta cell and liver, with decreased activity through loss of function mutation producing monogenic diabetes MODY 2, hexokinase 2 has two characteristics that suggest it is dysfunctional in insulin resistance and dysglycemia. One, insulin dependent expression, and two, it is stabilized to proteolysis at high glucose concentration. Glucose concentration is greater than about 7 millimolar. It is the latter process that causes hexokinase 2 linked unscheduled glycolysis. We know that metabolic dysfunction in early stage glycolysis is induced in vascular cells by hyperglycemia, and similar mechanisms may occur in skeletal muscle and adipose in the post-absorptive fasting phase. Characteristics of this were proposed in 2001. It involves increased glucose metabolism by the polyol pathway, increased flux of fructose 6-phosphate metabolism by the hexosamine pathway and increased flux of dihydroxyacetone phosphate DHAP metabolism to form increased diacyl glycerol de novo and activation of the protein kinase C pathway, and increased degradation of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate to form methylglyoxal in the dicarbonyl stress pathway. Now, there is very little glucose metabolized by the polyol pathway at vascular sites in skeletal muscle and adipose tissue. The remaining pathways are dependent on increased glucose metabolism by hexokinase. At sites of vascular complications development and in skeletal muscle and adipose tissue, there are only two forms of hexokinase, hexokinase 1 and hexokinase 2. Both are saturated in normal glycemic conditions. Key to initiation of metabolic dysfunction is stabilization of hexokinase 2 to proteolysis by high glucose concentration. We found this in human aortal endothelial cells incubated in high glucose concentration in vitro. Hexokinase 2 has a second active site in the C-terminal domain, wherein lies the degradation motif that binds heat shock conjugate 71, directing it to chaperone-mediated autophagy. Increased binding of glucose to hexokinase 2 in cytosolic hyperglycemia masks this site, stabilizing hexokinase 2 to proteolysis. Hexokinase 2 abundance and activity is increased up to twofold without increasing expression and activity of other glycolytic enzymes. This sends a wave of increased glycolytic intermediates through early stage glycolysis, driving all metabolic dysfunction. This is hexokinase 2 linked unscheduled glycolysis. The consequences of hexokinase 2 linked unscheduled glycolysis are increased flux of glucose metabolism, creating a wave of increased glycolytic intermediates. This stimulates hexosamine, protein kinase C, and dicarbonyl stress pathways. Increased glucose 6-phosphate induces detachment of hexokinase 2 from mitochondria, mitochondrial membrane polarization, and increased reactive oxygen species ROS formation. In 2001, it was proposed that increased ROS and oxidative stress is the initiator of metabolic dysfunction, but it is rather one step earlier, glucose-dependent stabilization of hexokinase 2 to degradation. And increased ROS is one of multiple downstream pathways of metabolic dysfunction activated by hexokinase 2 linked unscheduled glycolysis. A marker of hexokinase 2 linked unscheduled glycolysis is increased glycogen synthesis. Detachment of hexokinase 2 from mitochondria stimulates glycogen synthesis by metabolic channeling. All metabolic dysfunction stimulated by hexokinase 2 linked unscheduled glycolysis is preventable and treatable by glyoxylase 1, GLO1 inducer, transresveratrol plus asparatin combination, which I will mention later. Now, why do I call this unscheduled glycolysis? Normally, increased flux of glucose metabolism into tissues through early stage glycolysis occurs with increased activation and transcription of glycolytic enzymes. Increased activity of hexokinase 2, increased activity of phosphofructokinase and glyceraldehyde 3-phosphate dehydrogenase are produced by regulation by insulin. Increased activity of glucose 6-phosphate isomerase, aldolase, triose phosphate isomerase, TPI, and glyceraldehyde 3-phosphate dehydrogenase are increased by regulation by glucose 6-phosphate mondo A MLX. This is regulated or scheduled glycolysis. In hexokinase 2 linked unscheduled glycolysis, hexokinase 2 protein abundance is increased discretely, and all of this other regulation is dormant. Glycolytic flux increases, and there is a concomitant increase in glycolytic intermediates to abnormally high levels. This is dysregulated or unscheduled glycolysis. To appreciate the mechanism of hexokinase 2 linked unscheduled glycolysis development, a mathematical model can be constructed as we have done here. We add in terms for hexokinase 1, hexokinase 2, and also terms for hexokinase 2 synthesis and hexokinase 2 degradation with inhibition by glucose with an inhibitor constant of 7.5 millimolar. The model is adapted from that of Koenig et al., and we produced the model and ran it in the open source software program called Capacity. Here is a brief overview of outcomes. 5 millimolar glucose is the control condition where hexokinase 2 concentration is 0.17 micromolar, glucose 6-phosphate concentration is 10 micromolar, fructose 6-phosphate concentration 5 micromolar, and the rate of methyl glyoxal production is 2.7 micromolar per hour. When we increase the glucose concentration to 20 millimolar and making no provision for stabilization of hexokinase 2 by glucose, nothing changes. The concentrations of hexokinase 2, glucose 6-phosphate, fructose 6-phosphate, and rate of production of methyl glyoxal remain unchanged. But when we build in at 20 millimolar glucose a term for stabilization of hexokinase 2 by glucose, then things change. Hexokinase 2 concentration increases 76%, glucose 6-phosphate concentration increases 50%, fructose 6-phosphate concentration increases 56%, and the rate of production of methyl glyoxal has gone up to 177%. So with unscheduled glycolysis, a wave of increased glycolytic intermediates develops related to the duration of hyperglycemia and turnover of hexokinase 2. So where does hexokinase 2 linked unscheduled glycolysis occur? As we have published previously, there is clinical evidence of hexokinase 2 expression and downstream metabolic dysfunction of hexokinase 2 linked unscheduled glycolysis at sites of vascular complications in diabetes. Increased reactive oxygen species, increased hexosamine and protein kinase C activity, and dicarbonyl stress. This likely contributes to the development of diabetic endothelial dysfunction, diabetic nephropathy, diabetic neuropathy, diabetic retinopathy, and diabetic embryopathy. But what is the involvement in insulin resistance? Now, is there evidence of downstream metabolic dysfunction of hexokinase 2 linked unscheduled glycolysis in skeletal muscle in insulin resistance? Indeed, there is clinical evidence and preclinical evidence. Increased reactive oxygen species in insulin-resistant human skeletal muscle, increased hexosamine pathway activity in skeletal muscle of patients with type 2 diabetes, increased protein kinase C activation in skeletal muscle and insulin resistance in the high-fat diet-fed mouse model, increased dicarbonyl stress, increased methyl glyoxal in human skeletal muscle and insulin resistance. But how about adipose tissue? Now, is there evidence of downstream metabolic dysfunction of hexokinase 2 driven unscheduled glycolysis in adipose tissue in insulin resistance? Firstly, hexokinase 2 accounts for 66% of hexokinase activity in adipose tissue. Dicarbonyl stress has been found in adipose tissue in the high-fat diet-fed mouse model of obesity, a twofold increase of methyl glyoxal. Mitochondrial dysfunction and increased ROS has been found in insulin resistance in adipose tissue. Protein kinase C epsilon activation contributes to impaired glucose tolerance in insulin resistance in adipose tissue. And there's increased glycogen deposition in adipose tissue in obesity. Hexokinase 2 linked unscheduled glycolysis may start in the post-absorptive phase in adipose tissue and release increased methyl glyoxal and low-grade inflammatory mediators and decreased adiponectin to skeletal muscle and liver, stimulating insulin resistance therein. But where do we look for the clinical driver of hexokinase 2 linked unscheduled glycolysis? The answer is we look at non-insulin mediated glucose uptake in the post-absorptive fasting phase, which is independent of insulin. In an elegant study by Gerald Olewski's lab of 1985, NIMGU was measured in healthy control subjects with normal insulin sensitivity and in patients with type 2 diabetes. NIMGU was measured by serum glucose clamped, insulin deficiency induced by infusion of somatostatin, and glucose disposal by tritiated thiamine infusion. The rate of NIMGU was increased about twofold in patients with type 2 diabetes and accounted for 75% of total glucose disposal in controls and 71% in patients with type 2 diabetes. The authors said, in the post-absorptive state, NIMGU is the major pathway for glucose disposal. Absolute basal disposal rates of NIMGU are higher in type 2 diabetic patients. Elevated basal rates of NIMGU in type 2 diabetic patients may play a role in the pathogenesis of the late complications of diabetes, but also insulin resistance. In a classic study by DeFronzo et al. Of 1985, insulin-stimulated whole body and leg glucose disposal was measured in lean subjects, healthy controls, and patients with type 2 diabetes. We're interested in the baseline disposal before insulin clamp increase was initiated. See the red square and the expansion. The authors stated the total amount of glucose taken up by the leg tissues in the basal state was significantly greater in the diabetic patients than in controls, 1.7 versus 1.1 milligrams per kilogram leg weight per minute. This is an increase of 55% and is a mass action effect related to the increased fasting plasma glucose concentration increasing from 5.5 to 8.8 millimolar. So this is evidence of increased non-insulin mediated glucose uptake in skeletal muscle of the leg in lean patients with type 2 diabetes, which likely drives hexokinase 2 linked unscheduled glycolysis. Clinical data from one of the most expert and experienced investigators in the field, Professor Ralph DeFronzo. Now let's consider if hexokinase 2 linked unscheduled glycolysis can explain functionally genomic studies in mice of insulin resistance. Firstly, overexpression of GLUT1 in skeletal muscle of mice induced impairment of insulin-responsive glucose uptake. Transgenic mice with skeletal muscle overexpression of GLUT1 had a 3 to 4-fold increase in basal glucose uptake and no stimulation of glucose uptake by insulin relative to wild-type control mice. Wild-type controls had a 2 to 3-fold increase in glucose uptake in response to insulin. Impaired insulin-stimulated glucose uptake was linked to impaired recruitment of the GLUT4 glucose transporter, a common feature of insulin resistance, and mediated by increased hexosamine activity. We interpret this through the hexokinase 2 linked unscheduled glycolysis hypothesis that overexpression of GLUT1 increases in situ rate of uptake of glucose in the fasting state, mimicking the effect of increased fasting plasma glucose with normal GLUT1 expression. And increased hexosamine pathway activity is an expected consequence of hexokinase 2 linked unscheduled glycolysis. We may conclude that increased glucose uptake in the fasting phase may drive the development of impaired GLUT4 responsiveness to insulin by hexokinase 2 linked unscheduled glycolysis. Secondly, partial knockdown of hexokinase 2 in mice improved glucose tolerance in the late stage of a glucose challenge. Heterozygous hexokinase 2 knockout mice had low plasma glucose and insulin concentrations at 60 minutes after glucose challenge compared to wild-type controls in a glucose tolerance test. We interpret this as lower basal levels of hexokinase 2 providing resistance to unscheduled glycolysis, noting that the benefits of decreased hexokinase 2 in the glucose tolerance test are expected at the late stage because glucose produces a time-dependent increase in hexokinase 2 protein abundance linked to metabolic dysfunction. We conclude that low basal levels of hexokinase 2 may provide decreased risk of insulin resistance, an interesting prediction to explore now clinically. Thirdly, overexpression of hexokinase 2 in skeletal muscle of mice impaired uptake of glucose on a high-fat diet in hyperinsulinemic euglycemic clamp studies. Mice overexpressing hexokinase 2 in skeletal muscle on a high-fat diet had a 60 to 70% decrease of glucose uptake compared to standard diet feeding, whereas in wild-type controls there was no change. Glucose 6-phosphate concentration in skeletal muscle was increased in the sedentary state of the transgenic mice with the high-fat diet feeding. We interpret this as hexokinase 2 linked unscheduled glycolysis in the fasting state of the hexokinase 2 transgenic mice with a high-fat diet led to impairment of insulin-stimulated glucose uptake by muscle. We conclude that high basal levels of hexokinase 2 may provide susceptibility to unscheduled glycolysis and insulin resistance, a further interesting prediction to explore now clinically. So what predictions from the hexokinase 2 linked unscheduled glycolysis hypothesis can we make for the clinical setting? Prediction 1. Increased fasting plasma glucose concentration is an expected risk predictor for the development of insulin resistance and type 2 diabetes. Well, it's been observed that increased fasting plasma glucose measured about 3 years apart is a risk predictor for the development of increased insulin resistance and type 2 diabetes, whereas change in 2-hour plasma glucose in an oral glucose tolerance test was not. So this is an important prediction that fits with the hexokinase 2 linked unscheduled glycolysis hypothesis. Prediction 2. Insulin resistance is an expected characteristic of subjects with increased fasting plasma glucose, including patients with type 1 diabetes. DeFronzo et al. Found that insulin resistance was a prominent feature of patients with type 1 diabetes, linked mainly to insulin resistance in peripheral tissues, particularly skeletal muscle. Hitherto, there has been no explanation proposed for this, so hexokinase 2 unscheduled glycolysis hypothesis now provides a working explanation. But how can this improved insight guide future improved treatment for insulin resistance? This relates to glyoxylase 1 inducers for treatment of insulin resistance. Initially developed to treat increased methylglyoxal or dicarbonyl stress, they now appear to be insulin sensitizers. The HATFF or HATSOF study clinical trial of an inducer of glyoxylase 1, GLO1, a dietary supplement optimized to increased expression of GLO1 by activation of transcription factor NRF2, corrected insulin resistance in overweight and obese subjects. This is why it may work. Firstly, genetic activation of NRF2 in skeletal muscle by partial knockdown of the antagonist protein KEAP1 prevented insulin resistance in the high-fat diet-fed mouse model of insulin resistance. Secondly, increased methylglyoxal dicarbonyl stress is a potent inducer of thyrodoxine interacting protein, or TIC-SNP, a mediator of insulin resistance. GLO1 inducer corrected dicarbonyl stress in overweight and obese subjects in the HATSOF study, where response is correlated with decreased TIC-SNP. Thirdly, glyoxylase 1 inducer also increases expression of glucose-6-phosphate dehydrogenase, decreasing glucose-6-phosphate and decreasing MONDO-A MLX glucose-6-phosphate dependent expression of hexokinase 2. So it takes down hexokinase 2 expression and corrects hexokinase 2 mediated unscheduled glycolysis. So in summary, unscheduled glycolysis is increased glycolytic flux without concomitant increase in expression and activity of downstream glycolytic enzymes, increasing steady-state levels of glycolytic intermediates. A common cause is hexokinase 2 stabilization to proteolysis by high glucose concentration. In conclusion, I'd like to say that hexokinase 2 linked unscheduled glycolysis likely develops in skeletal muscle and adipose tissue in impaired fasting glucose and initiates processes leading to oxidative stress, activation of protein kinase C, hexosamine and dicarbonyl stress pathways, and development of insulin resistance. Hexokinase 2 linked unscheduled glycolysis hypothesis explains outcomes of functional genomics of GLUT1 and hexokinase 2 in insulin resistant studies of mice. The hypothesis explains why fasting plasma glucose is a risk predictor of insulin resistance and why insulin resistance is common in patients with type 1 diabetes. Hexokinase 2 linked unscheduled glycolysis is likely linked to increased non-insulin mediated glucose uptake in dysglycemia initiated in skeletal muscle and/or adipose tissue. Our hypothesis opens a new field of insulin sensitizers, glyoxylase 1 inducers. Insulin resistance is now a malady with a mechanism. Finally, I'd like to thank the investigator team, my long-standing co-workers Dr. Ming-Sang Su and Professor Naila Rabbani in Qatar, and I thank the Qatar Foundation and Qatar University for funding our research. And I thank you for your attention and interest. Thank you so much.